Welcome to the Ticktective podcast and video series, a program of the Bay Area Lyme Foundation, where our mission is to make Lyme disease easy to diagnose and simple to cure. I'm your host, Leah Gertner, Director of Education and Outreach. Lyme disease is the most common vector-borne disease in the USA and is a global issue. This show includes interviews with the researchers we fund, as well as other interesting people in the Lyme scientific community. We are a nonprofit foundation based in Silicon Valley. Thanks to a generous grant that covers all of our overhead, 100% of all donations go directly to support research and prevention programs. You can find out more or donate at bayarealyme.org. Dan Salkeld, PhD, is a research scientist in the Department of Biology at Colorado State University. His field research has focused on plague and Lyme disease. Dr. Salkeld has studied the ecology and epidemiology of Lyme and other emerging tick-borne pathogens, such as Borrelia miyamotoi. He has almost 50 publications along with scientists such as Dr. Robert Lane at UC Berkeley, Dr. Nate Nieto of Northern Arizona University, Dr. Eric Lambin at Stanford, and the Vector-Borne Disease section at the California Department of Public Health. The tick ecology work that you and Dr. Nieto, who we recently lost, the work that you guys have done has been pivotal to our nation's understanding of the tick-borne disease epidemic. Your team has found ticks and tick-borne pathogens where no one ever has before, and especially here on the West Coast. How did you get into this line of work?
Slowly. I was interested in zoonoses, so any pathogens that transmit from wildlife to humans. And my background is as an ecologist, so much more in the wildlife area. A chance came up to work with Dr. Lane at UC Berkeley a few years back, and that was my introduction to tick-borne diseases. But I had been working on plague in Colorado beforehand, and other wildlife pathogens before that.
And have you ever been bitten by a tick?
So many times. So I got bitten by a tick here in Colorado, just walking with a colleague last week. We were discussing One Health and decided to do it outside to minimize risk of COVID. And I came home with a Rocky Mountain wood tick. I've been bitten by heaps of ticks in research before I ever started looking at ticks themselves. And then it has been an occupational hazard ever since.
Did you test the tick that you found on you?
Yeah, slowly. So I would send them to the lab when we were just doing the surveillance anyway. It never occurred to me that I should tell them to look at this one stat. But nothing has ever come up positive so far.
Good. You have found ticks in places where we thought we were safe, like redwood forests and beaches. And you actually have a new publication that just came out. I saw it featured on NBC News. Can you share the highlights?
Yeah, I just find it my duty to ruin everybody's pastimes in the outdoors. So, for a long time, we've thought of redwoods as being a tick-free environment. And ticks are definitely in lower abundance there, but we do find them with some regularity. And then more recently, we've been looking in that coastal habitat, like coastal scrub, or it's called chaparral or coastal prairie. And we've been investigating ticks there too. And we've been finding them in big numbers in the winter. And we've been finding them infected with a whole range of different pathogens, including Lyme disease bacterium and some of their relatives.
Yeah, I read that in one spot or maybe multiple spots, up to 1/3 of the ticks that you sampled were carrying pathogens.
Yeah, so that's when we look at all these different pathogens in one go. So traditionally, and we've done this many times ourselves, when you publish a paper, you focus on one pathogen. So, for example, you look at Lyme disease bacterium, or you might look at Anaplasma phagocytophilum, or something like that. And you generally present your results in the context of just that one bacterial species. And we kind of started accruing the results and looking at Borrelia burgdorferi that causes Lyme, Borrelia miyamotoi that causes a different fever, and Anaplasma too. And it suddenly occurred to us that when you look at all three, then the risk of a tick-borne disease gets substantially higher because there's this cumulative risk as you look at more and more pathogens. And so some places it goes to 6% for Lyme disease, and then you add another 5% for miyamotoi, and then you add another 4% for Anaplasma, and you are suddenly looking at a different kind of sphere of tick-borne disease risk than you had if you looked at only one.
Right. So somehow we need to get that information to California Public Health, to government agencies, to doctors and healthcare practitioners? How do you think that information could translate?
I think, yeah, I think it's especially in somewhere like California where we've traditionally considered tick-borne disease to be quite low, especially compared to the upper Midwest or the Northeast of the United States. It's definitely worth thinking about this by diversity of pathogens out there. And that, especially when, if they're not really recognized or understood, we tend to underestimate their presence in the first place. So I guess just considering that there's going to be more than a minimal Lyme disease risk is a good start.
So in that vein, you, along with the Bay Area Lyme Foundation, put together the Citizen Science Project, which was a huge success. I'll read a little bit about it here. Citizens around the US sent in over 16,000 ticks in a little over a year, almost 2 years. And your team identified and tested the ticks for several tick-borne pathogens. The citizen science study was the first to find ticks capable of carrying Lyme disease in 83 US counties were previously undetected. And in 2018, I saw Nate Nieto did an NPR interview, and he said, we budgeted to receive 2,400 ticks. Then all of a sudden we were slammed with 16,000 ticks. And the massive response shows how hungry the public is for information on the ticks that bite them. It also fed scientists an unexpectedly large data set and information on ticks from 49 states plus Puerto Rico. And I'm wondering if you can talk about that work and some of the highlights that came out of that citizen science project.
Yeah, this was a fantastic experience. And I think the idea came from the Bay Area Lyme Foundation. So we're really grateful for the idea as well as the funding that allowed us to do this. And like you said, the idea was probably that we would look at ticks sent in from people in California, and about 2,000 people were what we were expecting. And it just took off overwhelmingly at times, such that we got ticks from every state bar Alaska, and Alaska sent things in. They just tended to be spiders or scabs. But yeah, we got them from all over. I think we're 21,000 ticks and counting still. Even when the website for submissions went down, people still sent in ticks because they had got that information. And so we are still submitting manuscripts on new data submissions. But it's been fantastic. It's opened our eyes in terms of working out where and when people are exposed. One of the major things being that often ticks are sent by people from regions outside of the kind of understood geographical distribution for the ticks. But people are traveling and moving around, and so are their dogs, and so are birds. And so just to be aware that people are exposed to ticks outside of textbook defined geographies has been quite an amazing insight.
So going back to the beach study, I guess there wouldn't really be an easy way to search for ticks in the sand. I mean, where would you start? But can we definitively say they're not in the sand, or it's just no one's really looked?
So I don't think they're in the sand. It's not, well, especially if you have tidal zones, there's nothing for it to survive. But you can find ticks on the vegetation almost directly abutting the beach. And what we've been looking for, it's mainly like those coastal walks in California that everyone loves to go on, that are around the cliffs that go through chaparral and coastal scrub.
And you also had a publication that proved the prevalence in California of a lesser known strain of Borrelia, like we talked about before, Borrelia miyamotoi. And you identified some of its previously unidentified rodent vectors. I know the first known human case of Borrelia miyamotoi infection in the US was only discovered in 2013. And we do know that there are many people here who get sick and test negative on a standard Lyme test. And I'm wondering what you think the likelihood of it being a Borrelia miyamotoi infection is. What are the statistics around miyamotoi versus Borrelia, the standard Lyme pathogen Borrelia burgdorferi here, as far as your studies have shown?
I think it's a really interesting question, and it's definitely at that kind of intersection of what we know for sure, and then the kind of new interpretations of previously existing diagnostic tests, right? So what we found right around the Bay Area and kind of Portola Valley area was that we were finding as many ticks infected with Borrelia miyamotoi as Borrelia burgdorferi, the agent of Lyme disease. So we know that it's common in ticks, and we found Borrelia miyamotoi across Northern California, and in unpredictable ways. There's no way of saying whether it's more common or less common than the Lyme disease bacterium. They're found in kind of idiosyncratic distributions. But it certainly suggests that people are being exposed to it. And there's been work carried out strongly suggesting that people are being infected. And sometimes the issue is that the diagnostic tests might cross-react with other Borrelia bacteria. But in the case of miyamotoi, it's often with a bacterium that's only found up in the High Sierra. So that suggests people are being infected by miyamotoi in the Bay Area. And I think CDPH just put out a study last year looking at blood transfusion data. And they've shown rare cases of miyamotoi, but it wasn't a targeted surveillance. So it's definitely infecting people, but I think we still are only looking at a tip of an iceberg in terms of understanding how often and what the symptoms are in California.
And I know in California and on the West Coast, it's sometimes more common for people to be infected by a baby tick, by a nymph. Obviously, they're teeny, they're the size of a period at the end of a sentence. So they go unseen when they attach to people, and especially if it's, on your scalp or behind your knee or something like that. So I understand why people would get more infections from nymphs. But I think in your studies, you've also shown that the baby ticks, the nymphs, carry higher rates of pathogen and infectious agents. Can you talk a little bit about that?
So yeah, in California, it's kind of considered a standard pattern that the nymphs are more likely to have the Lyme infections than the adults. And so in terms of the life cycle of ticks, when they hatch as larvae, the first stage, they're uninfected with Borrelia burgdorferi. They don't have the Lyme disease agent. They have to feed on an infectious host, which in California is often the western gray squirrel. And if that western gray squirrel is infected, then I think roughly about 80 to 90% of the ticks that feed on it will become infected. And so when they molt, they become nymphs, and those are the ones that are infectious and can then pass on the infection to humans or other animals.
I know people are mesmerized with the complement protein in the Western fence lizard that cleans the ticks of the Lyme bacteria. So that is specific to the West Coast. And can you talk about that, please?
Yeah, so this is work done by Bob Lane at Berkeley, and it's amazing work. And like you said, it's amazing how it's infiltrated the California public. And if they know anything about Lyme disease, they know this, that the Western fence lizards have a factor in their blood that will destroy Lyme disease bacterium when the tick is feeding on them. And that probably has a major impact on the frequency of Lyme disease infections in California, because if a nymph that is infected feeds on a lizard, it will lose that Borrelia infection. And so you have adults ticks that have a much reduced prevalence of infection compared to the nymphal stages. So as they get older, ticks would tend, because they're feeding on lizards so frequently, will tend to lose the Lyme disease infection.
Which is another reason why nymphs are particularly dangerous for us here.
True in the Northeast as well. Because they're so small, they're hard to find, and you don't feel them, and you don't feel their bite. And then in California, at least, they're more often infected than the adult stages.
People often ask me why I think there are more ticks being discovered north, east, west, and south than there were before. I tell people climate change is a big factor, moving into more natural habitats as we expand our neighborhoods, and changing ecological factors all contribute to the expansion, as well as maybe more surveillance. But I'm wondering what you think and what you think the reasons might be.
I think you sounded really articulate and spot on, and I'm not going to say anything. No, I think it's absolutely that. So I think the tendency we always have to explain any change or emergence of disease is to focus on a single factor. And you say, oh, it's climate change, or you say, oh, it's travel, or whatever. And I think for tick-borne diseases, it gives a nice overview for all of these emerging infectious diseases that we hear more and more about, that it's often a combination of lots of different factors. So for sure, Lyme disease is responding to climate change. But if you look in the East Coast, it's also traveling south, and it's also traveling west, right, which wouldn't be explained by climate change. And there, it's things like different housing patterns. People are enjoying building houses and living in the woods, or suburbia that still has those kind of forested plots. It's reforestation in the northeast. It's different habits of people actually getting out to the outdoors and enjoying it all the time. And so, and surveillance, like you said, we're looking for them now that we're aware that they're moving. Then we look to see where they move, and not surprisingly, the data fulfills our predictions. So I think it's what makes it exciting as an ecologist that these things are so dynamic. It's not just let's describe ticks and tick ecology and we can put this all in a textbook and forget about it. It's constantly changing. And that leads to exciting discussions and disagreements, but it makes it a really academically interesting field.
So what do you think about the future of ticks in the United States?
I think things are moving. I think they're catching up with changes in habitat. I think we are moving them too. So in Colorado, we have ticks moving all the time. So 2 years ago, people started calling me to say they had Lone Star ticks in their backyards, which if you look at the CDC maps, should be 2 states away from Lone Star ticks. We've been finding American dog ticks in Colorado at increasing numbers. And again, if you look at CDC maps, the distribution, they're not supposed to be in the state, but we're finding them. And veterinarians are finding them and sending them into universities, and the CDPHE here, Colorado Department of Public Health and Environment is finding them. But these things are changing all the time. And I think there'll be some spots who gratefully will watch ticks recede, but I'm not sure that's going to be the majority of the country. And I think it's something we should all be aware of increasingly.
And what do you do for tick bite prevention?
I have a pair of field pants when I go out, whether I'm hiking or doing field work, and then I do careful tick checks at the end of the trail, and then we do them when we get home as well. And if we know we've been in ticky habitats, which is one of the conundrums, right? You don't always know, but if we do, things go in the dryer and we have showers, but it's not foolproof. Yeah, I have been in big trouble in my family for coming home from field work and bringing ticks home.
I know, having been at the Bay Area Lyme Foundation during the Citizen Science Project, that we received in the mail envelopes with ticks and scabs and things like that. I know there are some stories that you have. Can you share them with our listeners and viewers?
On both counts. So we, on the Citizen Science team, one of the questions was, where did you find the tick? And in our scientific heads, we are totally thinking geographically. Did you find it in woodland? Did you find it in a national park or a state park? But the receiving end of that was like, oh, where did I find the tick? I found it on this part of my body, and here are photos. And so I know some of the grad students working with Nate were like, this, we should not be exposed to this kind of information. This is not our pay grade. This is possibly, human, what's it called, personnel issues. And then also, once you become a tick person, I have colleagues here who will call me and say, I've got a bunch of ticks in my freezer for you. And I will tell them, I've never asked you for them. I've even gone to dinner with people, and the first thing they've done is plunk Ziploc bags full, like, of moving larvae where a female has obviously been caught and just given birth. So there are hundreds and hundreds of ticks. And they say, here's this for you. What are you having to eat? I'm like, there's a time and a place.
Right. Yes. I know. I field a lot of tick questions in my free time. So with the larvae, I know that because some ticks were sent to you as engorged and pregnant females, which usually it is female if it's engorged, right? Because the females are the ones who are more likely to have a blood meal. So I know that sometimes while waiting to be processed, they've released their eggs. And so you open it up and you've got 1,000 larvae. And in some of those, you've been able to test for pathogens, right? Have you found pathogens in larvae where we thought that larvae were born without pathogens before?
Yes, so that's one of the interesting spots about Borrelia miyamotoi, is that can be transferred from mother to offspring. And so larvae are really hard to find. Like we're not even sure the extent to which they bite people, because they're so tiny that the chances of finding them are really slim. Like if we collect them, you can just see them as a glint of slight brown on a cloth when they catch the sun. Like you can't even see them reading glasses or no. So Borrelia miyamotoi can be transmitted by larvae, but we are struggling to find good numbers. And so a couple of papers have come out recently, and including ours this morning or whatever it came out today, in which we have a single infected larva. And so it's really hard to make inference or understandings about what that means ecologically. It does mean that there's a risk, but we haven't found huge numbers of infections yet.
Have you found any new pathogens in larvae that we thought wasn't transmitted transovariously?
So the work so far that we've done often has focused on known human pathogens. So Borrelia miyamotoi, newly recognized as a human pathogen in 2013 in the States, is something we're investigating. So we haven't looked for new pathogens yet, but we have just started this last year doing surveillance of the virome. So looking at viral pathogens, and we don't know yet what that means in terms of human health, but it's a nice new avenue to look at of what are the viral diseases that are in California ticks and other tick populations too.
That's great. Scary, but great. And then wasn't there a tick in your citizen science study that you thought shouldn't carry a certain pathogen, but then it did?
Yeah. So the issue with those is sometimes we're finding pathogens that are known to be transmitted by a different tick species, but we find them in a one that is not considered a vector. So for example, we can find Lyme disease bacterium in American dog ticks, and we have found some of those before, as have others. I think so far most of the experimental investigations have found that those ticks don't actually transmit the pathogen. It's just that they're picking it up during a blood meal themselves. And so the consensus is that often these ticks are not vectors. But it also raises interesting questions. So we're looking at, sometimes we're finding a Borrelia species in the Lone Star tick, and we don't know yet whether that's Borrelia burgdorferi that causes Lyme disease or related Borrelia. It could be Borrelia Lone Stari or something. So that all requires sequencing, and then if you're going to be really diligent, doing experimental infections in animals of different tick life stages to find out what goes on.
And you found ambuloma ticks here where there weren't supposed to be any?
Yeah, we found six, I think overall six Amblyoma americanum Lone Star ticks in California. And the traditional explanation for that is that these are associated with travel. We didn't have great evidence from those people that submitted them whether they had traveled or not. And I know California Department of Public Health every now and then gets Lone Star ticks too. The issue with those ticks is they seem to be spreading from their traditional range, and we're finding them in Colorado where they shouldn't be too. So that might be one of those tick species that is really worth watching in terms of new geographies that it can be traveling to. And people might be playing a role in seeding new areas with that tick, like if they're bringing them home or transporting them with dogs. And who knows whether they'll be able to take off in new places.
And that is the tick that is responsible for the alpha-gal meat allergy, which is also becoming a little bit more common here.
Yeah, exactly.
Do you have any advice for anyone who might want to go into ecology of ticks for their studies or their profession?
I love it. I think it's a fantastic intersection of working with ecology, conservation, public health, and one almost has to second guess everything one learns and thinks, and the dogmas are challenged all the time. So as a field, I think it's really fascinating. And I think I like that applied aspect. It's not just ecology, but we're looking at things that have implications for human health and happiness and safety. So I find it really rewarding, and I love being out in the field.
Well, I'm glad that you're out in the field, and I did go with you once. So I appreciate that you're doing that field work for us and all the things that you have helped the Bay Area Lyme Foundation with in terms of showing our health departments what's here. So thank you.
No, thanks you, Leah, and thanks to the Bay Area Lyme Foundation, because it's been a really fantastic, I guess, half a decade now of doing these kind of studies.
Thank you so much for joining us on the Ticktective podcast and video series, Dr. Salkeld.
Thank you, Leah. It's been a pleasure.
Thank you for joining us for this episode of the Ticktective podcast and video series, a program of the Bay Area Lyme Foundation. You can find out more or donate at bayarealyme.org.